students, so today we will see about the plasma. So we will see what is the plasma. So plasma is a pale yellow color fluid component of the blood. So it is a pale yellow color fluid component of blood that holds the cellular elements of blood in separation. So it holds the cellular element of blood in separation. It is a pale yellow color in fluid component and it holds the cellular element of separation. And plasma is a fluid portion of the blood. So it is a fluid portion of the blood. Its normal volume is 35,000 ml. So its normal volume is 35,000 ml. It is mainly composed of water. So the plasma is mainly composed of water. The percentage of water is 91%. And other substance present in the plasma are organic substances and inorganic substances. So we will see what are the organic substances. Organic substances are Na+, Ka+, Ca++ ions, iron and copper. So these are the inorganic substances, and we will see what are the organic substances. Organic substances are the proteins, lipids, glucose, urea and creatinine. So these are the organic substances. So we will see what is a serum. So serum is nothing but a plasma without the clotting factors. So without the clotting factors, it is known as a serum. So it is a fluid that remains after allowing the blood. So the fluid that remains after allowing the blood, and the clot and the later removing the clot is called as a serum. So after removing the clot, it is known as a serum. And we will see what are the plasma proteins. So plasma proteins are nothing but the proteins present in the plasma. So plasma proteins are the proteins present in the plasma, and they continue about 7% of plasma made up of albumin, globulin, fibrinogen, prothombin. So they contains about 7% of plasma, and they are made up of albumin, globulin, fibrinogen and prothombin. So we will see the formation of plasma proteins. So the most of plasma proteins are synthesized in the liver. So these most plasma proteins are synthesized in liver, and the albumin, prothombin, fibrinogen are also synthesized in the liver. But globulin is formed by the reticuloendothelial cells. So these are synthesized in the liver, means albumin, prothombin, fibrinogen are synthesized in liver, but the globulin is formed by the reticuloendothelial cells, plasma cells and lymphocytes. And we will see what types are there in plasma proteins. So there are important plasma proteins. They are albumin, globulin, fibrinogen and prothombin. So we will see what is albumin, globulin, fibrinogen and prothombin. So albumin is a molecular weight of 66,000. So it is a molecular weight is about 66,000. It is produced in liver. So it is produced in liver. The plasma half life of albumin is 19 days, responsible for maintenance of osmotic pressure. So this albumin life cycle in plasma is 19 days, and it is mainly responsible for the osmotic pressure. So the main thing we should note that albumin will be maintains the osmotic pressure. So it is also helped in the transport of bilirubin, hormones and drugs. So it is also helps in the transport of bilirubin, hormones and drugs. Next we will see what are the globulins. So globulins has a molecular weight of ranging from 90,000 to 1,56,000. So it is the molecular weight is about 90,000 to 1,56,000. And the globulin include transport of proteins, lipoproteins and immunoglobulins. So these include transport of proteins, lipoproteins and immunoglobulins. There are we can see three factors. So these fractions are alpha globulin, beta globulin and gamma globulin. So these are the three factors. And we will see about the fibrinogen. So fibrinogen has a molecular weight of 3,40,000. So its molecular weight is about 3,40,000. It plays an important role in blood coagulation. So it plays an important role in blood coagulation. It is mainly responsible for viscosity of blood due to its asymmetrical shape. So it is mainly responsible for viscosity of blood due to asymmetrical shape. Fibrinogen is mainly responsible for viscosity of blood. And we will see what is the prothombin. Prothombin is an inactive precursor of thrombin. So it is inactive precursor of thrombin. So main prothombin is an inactive precursor of thrombin. Its plasma proteins with the molecular weight is about 68,700. So molecular weight is about 68,700, and the normal concentration of prothrombin in plasma is, so it has a normal concentration that is 15 mg per dl. So it is a normal concentration is 50 mg per deciliter, and it is formed in the liver with the help of vitamin K. So it is formed by the liver with the help of vitamin K. So with the help of vitamin K only the prothombins are formed in the liver. And we will see what are the functions of plasma proteins. So they are the main functions of plasma proteins are maintenance of colloidal osmotic pressure, maintenance of viscosity of blood, buffering action, protein reserve, immunity, blood clotting, transport. So we will see about the maintenance of colloidal osmotic pressure. So in colloidal osmotic pressure, the normal colloidal osmotic pressure in the plasma is 25 mmHg. So the normal osmotic colloidal pressure will be 25 mmHg, and the plasma proteins are responsible for this pressure. So the plasma proteins are the only responsible for this pressure. So the pressure is in the plasma is 25 mmHg, and the capillary wall is impermeable to the plasma proteins. So this capillary wall is impermeable to the plasma proteins, therefore water is drawn into the blood. The maintenance of colloidal osmotic pressure is to water is drawn into the blood. So we can see this colloidal osmotic pressure in albumin. So it is the functions in the plasma proteins, and we will see what is the maintenance of viscosity of the blood. So in viscosity of the blood is the flow 4 to 5 times more than viscous of water. So blood is 4 to 5 times viscous than the water, and the viscosity of the blood depends upon the shape and the size of proteins molecules in plasma. So it depends upon the shape and size protein molecules present in the plasma. So the viscosity of the blood will be depends upon the shape and size of the proteins molecules present in the plasma. And we will see what is the buffering action. So buffering action means plasma proteins act as a buffers. So in buffering actions, we can see in buffering actions the plasma proteins act as a buffers, and they maintain pH at 7.4 by accepting or donating H plus ion. So by the accepting or the donating H plus ion, it maintains the pH of the blood, and they are responsible for 15% of buffering capacity of the blood. So these are responsible for the 15% of buffering capacity of the blood. And the protein reservoirs. So protein reservoir are the plasma proteins present in the blood act as a reservoir. So we can see in the protein reservoir heading only, it acts as a reservoir of the proteins. So it acts as a reservoir of the proteins. They are especially helps during the starvation. So they are especially helps in the during the starvation. And the immunity. So we can see the immunity. Alpha globulins are the antibodies that protects the body against the invading microorganisms. So we can see gamma globulins in globulin. So these globulins act and protect as a body against the invading microorganisms. So the main function of immunity is gamma globulins are antibodies that protects the body against the invading microorganism. And we will see what is the blood clotting. So blood clotting means fibrinogen and prothombin are responsible for blood clotting. So in blood clotting, fibrinogen and prothombin are responsible for the blood clotting. So we have seen that in immunity, gamma globulins. So we have covered albumin in colloidal osmotic pressure, and globulin in immunity, and blood clotting, fibrinogen and prothombin. So these four types of plasma proteins are seen in functions of plasma protein. And next we will see about the transport in functions of plasma proteins. So transport means a plasma proteins transport hormones, drugs and metabolics by the forming loose bond with them. So these plasma proteins transport hormones during and metabolic by the forming loose bond with them. Next we will see separation of plasma proteins. So separation of plasma protein is done to study them and also diagnose disease. So this is the helpful to study them and also diagnose disease. So there are different methods to separation of plasma proteins. They are salt separation, paper electrophoresis, Coy's functional precipitation, and immunoelectrophoresis, and ultracentrifugation technique. So these are the different types of plasma separations. And next we will see what is the hypoproteinemia. So in the heading only we can see hypoproteinemia, decrease the level of plasma. Proteinemia means proteins means plasma, decrease the level of the plasma. So hyperproteinemia is a condition where a decrease in the level of plasma protein. So it is the condition where the decrease the level of plasma in proteins, and the plasma proteins levels decreased in prolonged starvation and malabsorption. So these plasma protein levels will be decreased in prolonged starvation and malabsorption. So they are also decreased in the liver and kidney disease. So these plasma proteins are also decreased in the liver and the kidney disease. So we will see how what in liver disease it causes. So in liver disease, plasma protein level disease due to the reduced synthesis. So due to the reduced synthesis only the liver disease in liver disease plasma proteins levels are decreases, and the normal globulin albumin ratio is received in liver disease. So the normal albumin globulin ratio is received in liver disease. And there and we'll see the leaner disease. So leaner disease means more amounts of proteins are lost in the urine. So when the more amount of the proteins are lost to the urine, it is known as a renal disease. So clinically edema develops in these patients due to the decrease in plasma. So we can see that edema develops in these patients and to decrease in the plasma. So osmotic pressure, which causes momentum of fluid in extracellular components. So this osmotic pressure which causes the momentum of fluid in the extravascular component. So in the extravascular component we can see the causes of momentum of fluid. And next we will see about Cryptogemia. So Cryptogemia and plasmapheresis. So Cryptogemia means it is a disease due to conjugatal deficiency of fibrinogen. So it is a disease due to the conjugatal deficiency of fibrinogen. It is characterized by defective blood clotting, and it is also characterized by defective blood clotting. Clinically, the symptoms includes brushing, bleeding in joints, nose, and excessive bleeding after injury. So when this fibrinogemia disease causes, we can see symptoms like bleeding, brushing joints, nose, and excessive bleeding when the injury occurs. Next we will see about the plasmapheresis. So plasmapheresis is nothing but a small experiment. So it was an experiment performed in the dogs by Whipple to determine the role of plasma proteins in diet. So to determine the role of plasma protein in the diet, a scientist performed an experiment is called as a plasmapheresis. So in this experiment, the whole blood is withdrawn and the cellular elements are injected back into the body. So in this experiment, the whole blood is withdrawn and the cellular elements are injected back into the body. The process is repeated until the plasma protein level falls to 4/100 gram per ml. So it is done here is a small mistake, 4 grams 100 ml. So 4 grams per 100 ml. So the plasma protein levels fall to 4 grams 100 ml, and therefore different diets are given and their effect on protein synthesis is studied. So after that after when we injected back into the body, the process is repeated in the plasma proteins to fall 4 gram per 100 ml. So after this the different diets are given and their effect on the protein synthesis is studied. And this technique of plasmapheresis is clinically used to remove excess of plasma proteins and certain antibodies harmful to the body. So it is clinically used to remove excess of plasma proteins and certain antibodies harmful to the body. Thank you friends. So please subscribe my channel. Any mistakes please comment me in the comment box. Thank you friends. Please subscribe and like my channel. Thank you friends.